Lecture number 11, Protein Chemistry, Proteomic Analysis by Dr. Hannes Rohr. We will speak about the second one technique, 2D liquid chromatography. In this technique, we can study protein complex without any gel electrophoresis. We will take the separated protein and denaturated or complex of denaturated protein and digest this protein with different proteases like trypsin. The protein will be fragmented to polypeptide fragment or different polypeptide. These polypeptide will be dissolved in a pH acidic with pH 3 to have a positive charge. Then they will subjected to two stages of liquid chromatography. First stage including ion exchange chromatography. The stationary phase will be cation exchange stationary phase, and the mobile phase will be salt gradient buffer or salt gradient solution, and the peptide will loaded on the cation exchange column and bind to this cation exchange column, and then successive elution by increasing salt concentration or salt gradient will lead to separation of polypeptides by regarding to their charge. Then the eluted polypeptide will come to the second column, which is reverse phase column. This reverse phase column containing stationary phase of silica gel bound to the hydrophobic hydrocarbon, 8 carbon atom or 18 carbon atom. This will interact with the hydrophobic group in the amino acid residue, and this binding will be separated. The polypeptide will be separated for second time regarding to their hydrophobicity. The separation here in the first regarding to the polypeptide charge, then or regarding to their isoelectric point, and then regarding to the hydrophobicity of each polypeptide. So we have two forces for separation. Then the eluted polypeptide will go to mass spectrometer to measure the molecular mass, and here we can use peptide fragmentation unit, including tandem mass spectroscopy. In this tandem mass spectroscopy, the polypeptide will be fragmented to many of polypeptides, smaller polypeptide, and each of these polypeptide will give the molecular mass, and we can compare the result or the obtained molecular masses with the computational or library with amino acid sequence, and by this way we can know the amino acid sequence of each polypeptide and we can compare also with the genomic sequence in DNA in the same organism or the same cell. Here the reverse phase column, this polypeptide enter to the column, and in the mobile phase, and the hydrophobic foot of the polypeptide will be absorbed in the hydrophobic surface of the reverse phase material, stationary phase, and then it will be absorbed in the stationary phase till organic modifier concentration come to the critical concentration to make desorption for the polypeptide for the stationary phase. This is liquid chromatography mass spectroscopy. If we have a protein mixture of A and B and C, and then we digest it with proteases, the proteases is specialized or specific, have a specific site to cut, and by this way we can have a lot of polypeptides. These polypeptides can be fractionated by liquid chromatography, by HPLC, and then separated by mass spectroscopy. This mass spectroscopy, this is we have the the mass spectrogram, and we can analyze it in by 5 ions and compare with the database, and we have 5 peptide sequence, and this 5 peptide sequence we can predict the fragment sequence of all peptide, and then we identify the protein. To understand this, we will go to the next slide. We have two ways for identify a protein. First, we will take the protein, digest it with the protease, and we have a lot of polypeptide. Then we go to tandem mass spectroscopy. Then we can compare the mass spectroscopy coming data by the database, and by this way we can identify the protein. Or we can go to the peptide mass fingerprint to compare directly with theoretical fingerprint that we have, and by this way also we can predict the protein or identify protein. Here the mass spectroscopy stage, we have 4 stages for mass spectroscopy. The first, introducing the sample to the mass spectrometer, then generate, and then we have chamber for generation of ion in the gas phase, including gas like helium or argon or neon. Then inert gas. The polypeptide will be fragmented to separate ion based different masses analyzer, and then we detect the ion. By this way, we can recognize the mass of the polypeptide. The protein sequence, the amino acid sequence. This is, we isolate the individual peptide fragment in second mass spectrometer and can obtain amino acid sequence. This is the mass analyzer, and this is collision cells, and this is mass analyzer 2. By this way, for example, we can compare the peptide sequence with protein database, and we can know the protein. For example, if we have molecular mass of 170, it means alanine and valine. When we have 204, it is alanine, valine, alanine. This is the tripeptide. And so by this way, cumulative mass, we can predict the amino acid sequence. Of course, this protein identification by peptide mass fingerprinting. Here we use mass spectrophotometer to measure the mass of proteolytic fragment. After protein digestion, we have a lot of polypeptide, and we can buzz them after separation for mass spectrophotometer. The mass spectrophotometer will measure their molecular mass. Then identification is done by matching the measured peptide mass to corresponding peptide mass from protein or nucleotide sequence database. Mass spectrophotometer or mass spectrometry will be shown in this slide. This is a protein complex, and then digest with trypsin. Then we have a lot of polypeptide. Each polypeptide will come to the ion source or ion, and then these ions will be charged with ions, and then the polypeptide will go to the detector with a speed regarding to their mass through the mass analyzer. Then we can compare the molecular mass with the protein database. From this we can know the molecular mass, and we can the separation of the molecule based on mass charge ratio, and by this way we can identify the protein. Here, protein identification by mass spectrometry or spectrophotometry. We can take the protein complex from gel electrophoresis. We have one band. We can get this spot and removed and take the protein and fragment it by trypsin or by any protease. And then this polypeptide mixture or polypeptide fragment will go to spectrum fragment generation by spectrophotometer, and then from the spectrum, this spectrum, we can match it with a library. This library could be sequenced by artificial trypsinated proteins, and these are give artificial spectra. This artificial spectra build up a protein library. We can match this protein library to predict or to identify the original spot or original protein complex that we separate by gel electrophoresis. Isotope coded affinity technique or tag. It is quantitative method to identify protein. In this method, we labeling a protein sample with heavy or light reagent. This reagent containing affinity tag and heavy or light isotope. Blue color is chemically reactive group from a covalent bond to the protein peptide like sulfhydryl group. The yellow color isotope labeled linker, heavy or light depending on which isotope we used. Then affinity tag enable the protein peptide bearing isotope coded affinity tag to be isolated by affinity chromatography in a single step. Example for this isotope coded affinity tag reagent. This is biotin affinity tag. This is bind to streptavidin agarose resin. This is the ligand or the tag, and it is connected with a linker with heavy version with deuterium or light version with hydrogen. This is with a star, could be deuterium or hydrogen atom. And then this thiol group here with blue color, this is bind to cysteine. And if we have cysteine residue in the protein, it is bind to it. And by this way, when we go to affinity chromatography, the biotin will bind to the stationary phase, and of course the thiol group bind to the protein, and by this way we labeling the protein and isolate it with isotope affinity or coded affinity tag. Here we have different. We mix the isotope labeling tag or coded tag with the protein, and then make proteolysis with trypsin. So we have different proteins or different polypeptide. They can be isolated on streptavidin beds, and all of them will go except the that having the ligand or tag, and by this way we can identify it by mass spectrophotometry or by mass spectrophotometry by fragmentation, and by this way we can detect the can make amino acid sequencing or also identify the protein. The advantage of this method, it is estimate relative protein level between sample with the response level with accuracy. about 10%, and can be used on a complex mixture of protein with and it is a cysteine specific label, reduced sample complexity, and the peptides can be sequenced directly by if tandem mass spectroscopy is used. The yield and the disadvantage, the yield is not specific, it is not a specific method, and it is a slight chromatography differences, and it is very expensive, and the tag fragmentation sometimes, and the meaning of relative quantification information, and the no presence of cysteine residue is cannot we can if we have no cysteine residue in the polypeptide, we cannot use this technique. Thank you for your